Okay. Hello, everyone, and good afternoon. Thanks for joining us today. I hope everyone is well and safe. It's my pleasure to welcome you to this exclusive event, the Inscopix European Neural Circuits webinar. My name is Fabrizio Sizia, and I am the Regional Sales Manager for Europe at Inscopix. In the past few months, the vast majority of laboratories around the globe have either been shut down or opened with reduced access due to COVID-19 pandemic. Although I would assume that with the relaxation of many lockdown restrictions in many countries, life in your labs is finally resuming back to normal. But at Inscopix, we have never stopped promoting science. And in the past weeks, we have launched a series of seminars called Insight Webinars. Our European team thought it was also time to promote science from a more Eurocentric point of view. And we came up with today's Inscopix European Neural Circuits webinar. And I'm very excited today to have two great speakers with us. We'll host Dr. Yaniv Ziv from the Weizmann Institute in Israel and Professor Jonathan Whitlock from the NTNU Trondheim in Norway, who are shortly going to give a 25-minute presentation each about the most recent findings. After each talk, there will be a panel discussion where whoever is attending this webinar can address questions to the speakers. There is a Q&A window at the bottom of this page where you can type questions at any time. We will collect the questions, and depending on the time left available at the end, we will do our best to prioritize the questions and address them to the speakers. Well, before kicking off with the scientific session, I just wanted to pass the shared screen to Mark Koch, the sales manager for Southern Europe and Middle East, that will give a brief introduction about Inscopix. Mark?
Yes, Fabrizio, I will start sharing my screen. Thank you. All right. Good afternoon and welcome, everyone. Thank you for joining us this afternoon. As Fabrizio said, my name is Mark Koch and I'm sales manager at Inscopix. Before kicking off with the scientific session, I want to briefly introduce Inscopix and our Miniscope solution, specifically for those of you who are not familiar with us. So who we are. Founded in 2011 as a spin-off from Stanford University, Inscopix is a neuroscience tech company based in Palo Alto, California. We're a dedicated team of neuroscientists who specialize in developing miniaturized microscope solutions for imaging and interpreting large-scale neural activity during natural behavior. What we do, our core innovation showed here, the miniature microscope or Miniscope. You're all familiar with the standard epifluorescence benchtop microscope as schematized here. Our Miniscope is a miniaturized version of this device. It includes all the key elements, excitation, emission, detection, and is optimized for calcium imaging. Its reduced size and its weight of less than 2 grams makes it perfectly suitable to be implemented on the head of small animals, providing single-cell resolution of neuronal or circuit activity in freely behaving animals. Our approach is to provide an end-to-end solution to study circuit neuroscience. It consists of 4 pillars: consumables and reagents, instrumentation, data acquisition and processing software. All these ingredients, alongside with our 4th pillar, scientific support, help our customers get them to successfully imaging, minimizing the setup and troubleshooting phases and focus on the science. We do offer a variety of tools in terms of reagents and accessories, such as our ready-to-image viruses, concentration optimized and validated for immediate use in various brain regions and species. Our green lenses with attached base plates, one less surgical step. They're available in straight or prism and different length configurations to target any brain region. Our unique express probes, the combination of the 2 latter items to even further streamline the workflow by combining integrated lenses coated with virus, currently available for dorsal striatum and recently D1, D2 specific. And that list will continue to expand in the coming future because these probes combine all surgical steps of the workflow to one single step, getting you to faster imaging with improved success rate and repeatability. Finally, our ProView kit for streamlining the implementation step and increase repeatability and success rate. Regarding the Miniscope systems, there are 2 platforms available. The first one is named the Envista and allows for GCamp imaging in freely moving animals. The second one is named Envoke. The system is equipped with 2 LEDs and enables simultaneous GCamp imaging and optogenetic manipulation. The Miniscopes are less than 2 grams in weight. They're equipped with an electrically tunable lens to enable electronic focusing and multiplane imaging, and can achieve close to 1 millimeter field of view. They can be complemented with our active commutator that avoids cable entangling during active behavior and allows for minimal supervision. Together with the hardware, we supply a dedicated software package composed of data acquisition and data processing software that allow identifying cells and extracting calcium transients from raw traces using a validated pipeline. The software also easily allows to synchronize the imaging data with other signals or videos, such as behavioral camera as shown here. Last but not least of our four-pillar solution, our support. Our field scientific consultants are here to support your research projects. They come and train you in your laboratory. They offer their expertise and support at every step of the process that leads to publication. Our European senior field scientific consultant, Jonathan Zapata, is online with us today. He'll go give a short presentation dedicated to the support and the FSC role at Inscopix after the talks, and is also available to answer any technical questions regarding our technology. So feel free to ask. To date, more than 100 labs around the world use the Inscopix technology. Thanks to our complete end-to-end solution, we're really proud for an important milestone that we've recently achieved. In fact, the Inscopix user community has published over 120 research articles pioneering neural circuit discoveries in areas including memory, sleep, brain dysfunction, and many others, and covering nearly all brain regions, which witnesses how our solution and support effectively work at driving circuit neuroscience breakthrough. Okay, I'll close the presentation of Inscopix and our solution now, and we'll start with some exciting science. A little reminder about the webinar's agenda. There'll be a panel discussion with questions after the speaker's talks. And before I introduce the first speaker, I just want to make sure that everyone listening knows how to interact with questions during the webinar. There is a Q&A window at the bottom of this page where you can type questions at any time. We'll collect the questions, and as Fabrizio said, we'll do our best to prioritize them and address them to the speakers or to our FSC. I will now stop sharing my screen. And Yaniv, you can start sharing your screen and unmute yourself. And during that time, I will give a short presentation of Dr. Yaniv Ziv, who's our first speaker. So Yaniv completed his PhD in neurobiology at the Weizmann Institute of Science in 2007 in the lab of Dr. Michal Schwartz, where he was interested in the role of immune cells in the broad context of neuroscience, including neurogenesis and brain disorders. He then joined the Department of Biology at Stanford University in the laboratory of Dr. Mark Schnitzer as a postdoctoral fellow, where he contributed to the work leading to the publication of the Miniature Microscope project and applied novel methods to study the long-term dynamics of CA1 place codes. He then joined the Department of Neurobiology at the Weizmann Institute in 2014 as a senior scientist, where he established his group, which focuses on the mechanisms and dynamics underlying long-term memory under both normal and pathological conditions. In today's talk, Yaniv will discuss how multiple maps of the same spatial context can stably coexist in the mouse hippocampus and the implication of these results to current notions in memory research. Yaniv, it's a real pleasure and honor to have you here with us today. Toda Rabba. We're really looking forward to your talk. So with no further ado, I will now mute myself and hand it over to you.
Thank you very much, Mark, for this very nice introduction, and thank you, Jonathan and Fabrizio and Jackie, for organizing this webinar. It's a pleasure to be here. So I will talk about some recent findings from our lab, where we found that multiple maps of the same spatial context or same spatial environment can stably coexist in the mouse hippocampus. So in our lab, we are interested in learning and long-term memory, and specifically, we are curious about how memory information is encoded and what happens to it over timescales that range from days to months. And we focus our work on the hippocampus and related cortical circuits that are important for learning and memory of places and events. And we rely in our work very much on optical imaging technologies, and specifically on time-lapse calcium imaging in freely behaving mice. I don't need to introduce this system for you. I'll just mention that for us, for the purpose of studying neural coding of long-term memory, this approach offers two critical advantages. The first is the ability to record from a fairly large population of neurons. We can now routinely record from many hundreds and even more than 1,000 cells simultaneously. And this is important because it allows us to perform both analysis on both the individual coding properties of individual neurons, and also to account for the coding properties of the population and the interactions between different subsets of cells within the population and within activity pattern. And we recently published a new approach that offers a new way to study the neural code by portraying and quantifying the internal structure of neuronal population activity. I will not talk about this today. The other key advantage of this approach for us is the ability to track the same neurons across multiple days in the calcium imaging data. And this is important because we can track how the coding properties of individual neurons change as a function of learning and as a function of the passage of time and so on. So this is an example of a typical data set we obtained in the lab. You can see here the same field of view within the same mouse imaged across 8 imaging sessions spanning 2 weeks. The different dots here represent the individual special footprints of individual neurons. And we also developed recently a technique that allows us to perform this cell registration routine in an automatic manner and in a way that evaluates the accuracy of each such decision of cell registration. And the project I'm going to show today actually showcase very nicely how these 2 advantages, being able to track the same neurons across multiple days and being able to record for many neurons, facilitated the discovery of an unintuitive and surprising phenomenon. And the project I'm going to present today is the result of a close collaboration between 3 different scientists with very different points of view, as you can see in this picture. Nitzan Geva, Liron Sheintuch, and Alon Rubin. And this project was also very recently published, so feel free to consult the paper for additional details I will not have time to cover in this talk. So this picture was taken, by the way, when we were all in Lisbon for the COSIGN meeting, where we presented this project for the first time. And the prevailing notion in memory research is that a given memory, like visiting Lisbon here for the first time for a COSIGN meeting, is supported by a specific pattern of neuronal activity, but specific neuronal activity, whereas other memories, other experiences are supported by different patterns of neuronal activity. Now, in the hippocampus, places form representation of space, and it is typically reported that when you record from places in a given environment and then move the animal to another environment, places typically remap, that is, they show different activity patterns and preferred firing location. And it is also typically reported that when you replace the animal to the same spatial context, the same spatial code is reused. So today, I will show you that even under the same, in the same physical environment, we can see place cell remapping without any apparent changes in sensory input, behavior, or motivational state. Now, the data that we analyzed in this project came from multiple experiments where we imaged the C1 of the hippocampus as mice ran back and forth along a linear track to collect water rewards at the end, just like you can see here in this movie. So the bottom panel shows you the mouse, whereas a miniature microscope runs back and forth along the linear track. We image C1 here on the top panel. So you can see the flashes of the calcium dynamics in the C1, and we marked 6 place cells here in different colors. And you can see here in the middle panel where the mouse is located along the linear track when each of these cells fires. And if you pay close attention, you can see that these cells respond not only to specific location, but also during specific running direction. Some cells prefer to fire in specific location during rightward to leftward running, and others during left to right running. And this is very typical for a one-dimensional familiar environment. Now, in this study, we had 27 freely behaving mice, from which we recorded over 8 imaging sessions. Each session was composed of the following structure. It had 5 trials of 3 minutes in which the mouse was running back and forth along the linear track, And in between trials, we had a 3-minute-long inter-trial interval in which we placed the mouse inside an opaque rest box or an opaque bucket just outside of the recording track. And what's also important to note is that we paid close attention to keep the all the conditions the same, and to place the mouse at each iteration in the same manner. Okay, so here you can see an example of 5 simultaneously recorded play cells that were recorded in one mouse. The blue line represents the trajectory of the mouse along the linear track, and the red and green dot shows you the position of the mouse along the linear track when each of these cells fired through either rightward or leftward running. The black line, black horizontal lines that you see here are the concatenation points between different trials. And I remind you that different trials are actually different visits to the environment. So in these 5 simultaneously recorded neurons, you can see that the same place field was retained in all 5 visits, all 5 trials to the same linear track. However, in this second mouse, we see something quite different. You can see that, again, these are 5 simultaneously recorded neurons as in the first mouse, but here you can see that trials 1, 2, and 4 had the same place fields, and they differed from trials 3 and 5, which had different place fields. Again, trials 1, 2, and 4 had the same place fields, and they differed from trials 3 and 5. And what I'm showing you here for 5 simultaneously recorded example cells, we can see also when we consider the entire population of place cells. So this matrix shows you the population vector correlation, which is a measure of similarity of the ensemble of place cells for all positions and all trials within this experiment, within this recording days. So each pixel here is the comparison between different positions and different trials. The comparison of the similarity of the representation. And what you can see here at the population level is, again, that trials 1 and 2 are of the same map, and trials 1 and 3 are of different maps. And what we see here at a single mouse, we can see when we pull together all the data from all the mice, and the population vector correlation are higher for the same map across all locations and across all cells, overall showing that population activity alternates coherently across all cells and locations. Now, what's also important to say is that we found these switches to occur only across trials, and it seems that they require disconnection from the environment. We quantified this in a number of ways. I'll show you a few. So here you can see the population vector correlation between a running window of 60 seconds and the average population vector of all trials within a given map. And what you can see is that when the population vector correlation was high for a given map, it was also very low for another map, and vice versa. And you can see this in all examples that I'm showing you here. And what's also important to note here is that the transitions between the different maps occur only across different trials. So these are individual examples, but we see the same thing when we look on the entire population and all the mice. And in fact, in more than 1,000 individual trials, we never saw switches within a trial. More than that, even if we look on individual frames of 50 milliseconds, we didn't see any fast switches or flickering between different maps. So for individual frames of 50 milliseconds, the PV correlation was high for the same map and was very low for different maps. And you see the same thing when you look on the joint distribution of the two. We see high density of data points with high correlation to the same map with low correlation to a different map, and near absence of data points with high correlation to a different map and low correlation to the same map. So no flickering of fast switches. Overall, the data suggests that switching between maps require disconnection from the environment. Now, as I mentioned at the beginning, one key advantage of this approach is the ability to record across many days, and we took advantage of this here, analyzing the recurrence of different maps over weeks. So what we found is that each of multiple maps can recur over weeks. What I'm showing you here is the matrix showing you the population vector correlation. Only this time, each entry in this matrix shows you the average for the average population vector correlation between for a given trial between all positions. So the reason this pixel here is dark orange, is red, is because the diagonal on this matrix is the average on this diagonal is high. So this is the population vector correlation across all positions. And likewise, the reason this pixel is blue is because the diagonal on this matrix is low. Overall, this is a 40 by 40 matrix because we had 5 trials per session and 8 imaging days. And what we see when we project this matrix on its leading eigenvectors is 3 very distinct separate clusters corresponding to different maps. And we can see it even nicer when we reorder the trials according to the different clusters or different maps. So you can see here the different trials were of 3 different clusters, 3 different maps that were very consistent within a cluster, within a map, and very different from the other maps. Now, also important to know that we found that alternations between representations involve both places and non-places. When we split the data to the part of the data that is composed from all the cells that met our statistical significant testing and we declare them as places versus the other cells, the non-places, we found them to behave very similarly, and we consider all pairs of trials. We see that the rate correlation between places and non-places is correlated both within session and across session, overall suggesting that these switches occur not only regarding the place code itself, but actually encompass all aspects of the episodic representation. Now, when people in the lab approached me with this finding of different maps for the same spatial environment, I was actually initially very skeptic about it, and I thought that maybe one map is the real map and another map is just noise. But actually, this is not what we found. We found that different maps, different spatial representations show similar high quality. For example, when we look on so each data point here is the average of different trials from the same animal. And when we quantify the fraction of significant places in one map, we found it is very similar to the fraction of significant places in another map throughout all our mice. And likewise, the degree to which the population is informative about position, the spatial information, was similar between different maps, and very different, much higher and very different than what you would get in the shuffle data. Overall, suggesting that the quality of different maps is similar and very good. So it's not like one map is good and the other is not. We also quantified other behavioral variables, and we didn't find any behavioral variability that could explain switching between the representations. For example, the fraction of the percentage of time that the mouse spends stationary, that is not running, was similar across maps. Also, the number of track traversal was similar across maps. The average speed was similar across maps. And we also didn't find anything in the gross statistical attributes of the neuronal activity that could explain the switching between representations. For example, the number of active cells was similar across maps, and also the number of events per cell. Now, as we did in our previous work, we could now take advantage of our ability to record over time and quantify the stability of the map over time. So what I'm showing you here is the population vector correlation between individual trials as a function of elapsed time. And we can see that the correlation decays with time as we consistent with our previous studies. But what we can do now is to separate this analysis for the different maps. And when we consider the stability within a given map, we see it's higher than what you would get for the naive calculation considering all maps. But what's important to see here is that this is also decaying with time. But even after 2 weeks, the stability of that map is much higher than what you would get across maps, which consistently stays around 0. Now, this finding of switches, all-around switching between different maps that occur also over days and weeks, raised the possibility that this switching in itself may play a role in driving these long-term dynamics in spatial representations that we discussed previously. And what we found is that indeed the probability of a given map to recur also over time also decays with time. So if you take 2 trials, you have higher probability for them to be of the same map if they are temporally proximal, if they are close in time, than if they are temporally distant. Okay, so overall, what this analysis suggests is that there are actually 2 components that drive the long-term dynamics of spatial representation. One is the changes in the map itself, and the other is a change in the probability of a given map to recur. Now, as I mentioned in the beginning, we found many mice, actually most mice displayed multiple distinct maps, like this mouse in this example. Again, these are the population vector correlation for individual trials over 3 different imaging days. But we also found a number of mice that had this phenotype had only one map, one stable map. And if we look on the distribution, we see in cyan about 1/3 of the mice had a single map, and in magenta here you see the distribution of those maps, of those mice that had more than 1 maps, the effective number of maps. But what's important is that we don't find any differences between either the quality of the code or the stability of the code between mice that had 1 map and mice that had multiple maps. So the degree to which the population is informative about position, spatial information, is not different between 1 map mice and multiple map mice, and also the stability of the representation is not different. So this suggests that there is an intact spatial code in mice with multiple maps. Now, how multiple maps are formed to begin with, we don't know. We are curious about the mechanism, but we can speculate. So, from a dynamical system point of view, the structure of connectivity of the network defines an energy landscape with attractors and separatrices between them. And you can think of the different maps that we find in this study as different attractor states. Now, this energy landscape dictate for each initial conditions of the network the specific attractor that will be associated with it. Now, what we consider is the following: is that during the process of learning, if there is low variability in initial conditions, the process of learning or familiarization with the environment will lead to the formation of 1 stable attractor, or 1 map for that matter. However, if there is relatively high variability in initial conditions throughout learning, this may lead to the formation of more than 1 stable attractor or more than 1 map. And this is an illustration, but this is something that we consider and would like to experimentally test soon. So just to summarize, I've shown you that there are multiple, we found multiple stable maps for the same spatial environments in mice. Spontaneous switching between representation we found is coherent across the entire neuronal population, places and non-places alike. We found that distinct representations are spatially informative and persistent over weeks. We suggest that distinct representations may result from variability in initial conditions of the network. This is something we would like to test test in the future. And I would like to end by discussing one question that is important and still left unanswered. And the question is, what is the meaning of multiple representations in terms of perception? So we consider two possibilities. One possibility is that multiple maps of the same environment reflect different perceptions under the same sensory experience, same sensory input, as often observed in famous optical illusions. In other words, the mouse will think or will perceive the environments, the same environment, as different under different maps. Or alternatively, these are actually the same perceptions under different neuronal representations, and this is consistent with an idea suggested by a recent study in bats that showed different hippocampal spatial codes under different sensory modalities. So this is also something that is still an open question. So with this, I'd like to finish. This is the lab in a pre-corona time, and I'd like to thank again the people who did the work. This work was led by Nitzan Geva, Liron Sheintuch, and Alon Rubin, and Yoav Rechavi and Adas Baumer also provided a lot of help. And this is it. Thank you for listening.
Thank you, Yaniv. It was very interesting talk. I think we have some questions for you. So I'm going to start asking you the first question from one of the person that is attending. So the first question is, do multiple space maps coexist just while the animal is performing goal-directed behavior? Or is it possible to observe them during simple spatial exploration, for example, during open field exploration?
So the question is, can we observe multiple maps for the same environment also in other contexts, such as open
field exploration?
This is an excellent question. We can say that we observed multiple maps for the same environment under different conditions that involved the early world, and it will be interesting to see whether we can see multiple maps also in cases where there is no reward or there's no goal-directed behavior. But we don't have a concrete data on that.
Okay, Yaniv, we have a second question for you. What do you think determines the variability in the initial conditions that led to different maps?
Right. So if I go back to this schematic, so the variability in initial conditions can be a result of so in our experiment we used the special environment with a lot of spatial, visual, and tactile cues. So it's a complex, rich environment that is complex and rich with sensory cues. And it is possible that in different iterations, different visits to the same environment when the mouse was learning, different sensory cues were used as a reference frame. It is also possible that there are differences in the inputs to the network itself. For example, it is possible that changes in the head direction signal that is conveyed to the hippocampus, combined with conflicting visual information, could lead to the formation of different maps. In real life situation, we think of situation like a change, like a dynamic environment that has some changes in the sensory cues that may lead to something like that. But that's of course a speculation.
Yaniv, I got another question for you. So it says, thanks for a nice talk. And then I would like to ask you how reliably you can track individual cells across time, and how many cells can you track across the whole experiment, especially given the limited Z resolution of 1P miniscopes.
Okay, so we can track. So Okay, so we can track, so within a given field of view, we see many hundreds of cells and even more than 1,000, and we can reliably track the same large number of cells over many sessions, many tens of sessions, and many days over multiple weeks without a deteriorating accuracy. We recently published a paper in which we, as I mentioned in the beginning, we introduced a method that allows us to perform this cell registration not only in an automatic manner, but also in a way that estimates the confidence we have for each such a decision. So, without getting into more details, I think we're pretty confident in our ability to see the same neurons across days. But maybe if I'm allowed one more minute, I think another question that is maybe relevant for our talk is that maybe what we see here is a reflection of our of a limited ability to see the same cells or anything in the any artifact in the imaging preparation. So I can talk about this a little bit. What we need to address this issue is to divide to first analyze data and find trials that are in different maps, and then based on this division, take again just the raw data for a given map and process the data separately and find the individual neurons separately for each individual map, and then combine them the way you see here using our cell registration approach. And when we do that, you see map 1 is in red, map 2 is in green, and the yellow is the overlay. We can see that those cells that were active in both maps are actually the same cells. There are no confusion here about their identity. And of course, as expected, we see that some cells responded to one map, some cells responded to other maps, and there are many cells that responded in both maps. But this procedure, one of several, also ruled out the possibility that it is a recording instability that drove these changes. And we also tested this using different cell detection algorithms, PCA and CNMF, PCI-CA and CNMF, and we got the same results.
Okay, thank you very much, Yaniv. Thank you again for the great talk. I think it was really, really good. And we'll have another opportunity for more questions after the second talk. So I think we should move forward in a matter of time to Jonathan Whitlock. So I think it's time for you, Jonathan, to unmute and start sharing your screen. In the meantime, I will introduce our next speaker. So Jonathan Whitlock. Jonathan completed his PhD in the laboratory of Dr. Mark Baer at Brown University and MIT, with his thesis work demonstrating that synaptic connections in the hippocampus grew stronger as a consequence of single trial learning in rats. He then moved to Trondheim in Norway for a postdoc with Professors Edvard and May-Britt Moser, Nobel laureates for the discovery of grid cells, and Jonathan's work focused on comparing navigational representation across parietal and entorhinal cortices. Following his postdoc, he established his group at the Kavli Institute for Systems Neuroscience at the NTNU in Trondheim, where he was appointed associate professor in medicine in 2019, and where his research focus is on unraveling how higher motor systems in cortex coordinate natural behavior and permit social learning. In today's talk, Jonathan will discuss his lab's attempts at uncovering the role of sensory motor mirror cells in mice during performance and observation of specific tasks. Jonathan, it's a pleasure and an honor to have you here with us today. We're really looking forward to your talk, and with no further ado, I will now hand it over to you.
Great. Well, thank you for the very kind introduction and for the invitation to share my group's recent efforts today. So the title of the work is Action Representation in Mouse Cortex. And it speaks to a general effort in my group to, we've spent more time in recent years trying to understand motor cognition, both from a first-person perspective and from a social perspective. And so this is our most recent efforts at getting at the neurobiology of social cognition, using the mouse as a model system. So the work in my group is mainly rooted in parietal cortex, and more recently we've spread out and recorded in frontal areas. So I'll just introduce parietal cortex, where it sits in humans and monkeys. So it's a multimodal association area. It sits on top of the occipital lobe, and it's behind somatosensory cortex, and combines inputs from these modalities as well as several others. And this is true both in the human. Here, when we look over at the macaque, you see it's situated similarly in front of occipital cortex, behind somatosensory cortex. And what's been really appreciated quite a bit more in the last 20 years is that there are discrete subregions of parietal cortex that combine different modalities of sensory information and combines them, and then exports this information, if you will, in a loop up to the frontal motor areas. So this is premotor cortex here, circled in red in the macaque, and it's the same situation in the human. There's the parietal frontal pathway. It's this very large, robust, bidirectional connection that exists between parietal and premotor areas. It facilitates, it subtends a variety of cognitive functions, but mainly what I'll be focusing on in terms of my work, but also in the talk today, is on motor functions. And more specifically, it's a specific type of cell, a very peculiar type of cell called a mirror neuron. So these are cells that fire when an individual performs or observes a specific action. They were first discovered in the premotor cortex in macaque monkeys, and later found in posterior parietal cortex, also in macaques, in the inferior parietal lobe, in the lab of Giacomo Rizzolatti. So I'm going to show a video now from Rizzolatti's lab, showing an example of a ventral premotor neuron that fires whenever the animal is grabbing a piece of food that it's going to eat. And I hope the audio is clear. Un altro così. Prima, ancora. Now it's the same cell. The animal's hands aren't moving. It's just watching this man holding a piece of food. Ancora. And there you go. There's your mirror neuron. So I think, still think that this type of tuning is absolutely remarkable. It's absolutely mysterious. And we know very little actually about the biological mechanism that makes this kind of tuning possible. So I really wanted to know, when I had first started my group, if mirror neurons were common across mammalian species, if you could see them in other animals, and really what is their biological mechanism? What are the neuronal projections? Ultimately, what are the cell types? The identities of the neurons that make this kind of thing possible. And if we want to do that, it's very advantageous if we can study this type of tuning in a simpler, smaller animal where there is lots of tools for studying the neurobiology, like mice or rats. So PPC, the posterior parietal cortex, as well as the frontal motor connection with PPC, is conserved over mammalian evolution. So here's drawing up, again, PPC in the back of the head of a human, between visual cortex and somatosensory cortex. And again, the projection up to frontal cortex is also conserved. So, and this is true down looking in macaque monkeys, galagos, tree shrews, really just in all mammals where it's been studied. You'll see it. So in rats and mice are no exception. They have the same kind of topography of visual and a sliver of PPC, and then somatosensory cortex. And again, motor cortex is up in front there. right? So then to get at action representation in mice, we were studying the posterior parietal cortex and frontal motor cortex during performance and observation of specific behavioral tasks. And the experimental end here was really spearheaded by Tucci Tombaz, who did the majority of the recordings in PPC. She was joined by Carolina Hovda, another PhD student who had recorded in M2, and Benjamin Dunn, who again was there from the beginning, writing and designing the analytical framework that we used really for the whole project. So, right, so we were doing, I should say they were doing just bulk injections of AAV1 expressing GCAM6M into the mouse PPC or the mouse, frontal mouse M2. And we were using the prisms so that we could implant them into the brain and look at deep and superficial layers. And of course, the Inscopix mini scopes were used for imaging lots of cells at a time. So the two tasks that we use, one is an open field task where really the item of interest here is when the animal's in the running wheel, because they're naturally inclined to do this. They like running on the running wheel. They'll do it in their home cages all day, and they would do it in this arena, no problem. They could run, clockwise or counterclockwise as they liked. And we would have an observer animal that was head fixed, that had been habituated to being head fixed and sitting in a tube just about 10 centimeters away from the running wheel in this case. The animals were siblings. They were raised together, and we almost out of happenstance, we ended up using entirely females for the study. We had used a mixed bag of males and females, but in the end, the animals for the data sets were all female. Right, so there's the open field wheel running task, and then we also used a pellet reaching task, again, where the animal is freely behaving. It can do a variety of behaviors, like rearing, turning, grooming, and of course, reaching for food. So pellets of food were dropped down from a container up above, and the animal could reach in through a hole and grab the food. And again, the observer is watching about 10 centimeters away, so quite close. And this is just an example of an open field of view, similar to what Yaniv had shown, but this is in cortex through a prism. So we're looking at deep layers here. Each of these arrows indicates just example cells from this particular recording. And it demonstrates that cells were active during both performance and observation conditions. So just to show you, just to give you a point-by-point sort of walkthrough of the pellet reaching test. This is an earlier version where we were still using forceps. We ended up getting rid of those, but this is an early version. It encapsulates the sequence. So the demonstrator has to turn away from the hole for food to be placed there, and the animal just reaches through and gets it. And here, just off camera, is a second mouse, and they're both wearing mini scopes. The observer is here just a few centimeters away, and there we have another camera trained on the face of the observer. Here she was squirming a little bit as food was delivered because she can't get the food. The animal's eating the food right in front of her, and she can't get it. And so, sometimes the animals would fidget. So we had to mark that and pay attention to it. Right. So here is the calcium imaging from the demonstrator animal. This is after CNMFE processing. This is the output of it that's shown here. So that's how the cells are rendered. And we had meticulous hand labeling of the animal's behavior. This was before we had nice algorithms like DeepLabCut or other ones that are now becoming more freely available. So we had to do everything by hand here, and this was inspected by another lab mate to make sure that everything is labeled evenly across animals. Right, and then this was all synchronized with the neural recordings. So shown in blue is the actual calcium traces that were recorded, and shown in red is the deconvolved firing rate estimate. This is using FOOPSI, the deconvolution algorithm FOOPSI, but since then we had switched to OASIS. All right, so that's the basic paradigm. And we found in PPC that there were cells that were tuned to all of the different behaviors we considered. And you would typically see them all, within a field of view. We didn't see any clustering or particular order to the distribution of the tuning of the cells. They were just kind of scattered, right? So this is a field of view showing deep parietal cortex, and the neurons are color-coded based on their behavioral correlates. So we quantified tuning to the nose poke, grasping for food, eating food, grooming, turning right, turning left, or rearing up. And in both parietal cortex and in M2, we found was about half the cells were stably tuned to these various behaviors we considered in both areas. Parietal cortex was dominated by rearing and turning movements. M2 also had strong representation for turning, but also about a quarter of the cells were tuned to when the animals were grabbing food out of the hole and eating it. So the action, the tuning to these specific behaviors was stable in PPC. So here's an example of a cell that its firing rate was significantly above shuffle distribution across 2 sessions. So that's why we labeled it a nose poke cell, because it was most active during the nose poke condition for 2 sessions. And so if you look within 1 session here, each row corresponds to an individual trial when the animal was poking its nose. And shown here at 0 is the onset of the nose poke itself. And so this cell, you can see in the average that relative to before and after the nose poke, the cell is quite active during the nose poke. And if you look below now, this is the population average for all animals that had nose poke cells. And each row here corresponds to the mean activity of the neuron over a given recording session for whenever the animal did a nose poke. And again, you see you have a nice flat baseline, and the nose poke cells were active during the nose poke. And this was the case for all 7 behaviors that we considered in the test. So we had cells for grasping, for eating, for grooming, turning counterclockwise, and rearing up. And again, these are the population averages shown beneath, and the average traces for this are shown with intensity shown on the y-axis, or the estimated firing rate rather is shown on the y-axis. Right. This is also true for M2. We had widespread stable correlates for performed behaviors that we considered in the task. However, after a couple of years of really quite steady effort, it started to become clear that the observation-related correlates for the corresponding behaviors weren't there. We weren't finding them. So this is an example of a cell that fired really beautifully when the animal would reach and eat food. The cell was recorded in PPC. Great. So then you take the animal out of being head fixed. It gets placed in the demonstrator box. And now the observer, the performer here becomes the observer. You take the animal out of the box, fix the head, put it in the tube it's watching. And whenever the demonstrator would grasp, we did not see neural activity. Okay, so then you take the animal back out, put it back in the box. It does the task again. We see this nice stable tuning. You put it back in the observation condition, and the cell wouldn't discharge. Right, and so this is true for really all the behaviors we considered. So this is turning counterclockwise. This is for rearing, and again, in all these cases, you see quite nice behaviorally locked activity, fairly locked activity during the free behavior, but nothing during observation. Again, performance, not during observation. And these are just correlation matrices that we showed. We calculated and made to compare the stability of the tuning of the cells for their behavioral preferences across conditions. So each one of these rows here corresponds to one of the different behaviors we looked at, and the color coding is it's the Pearson's correlate of that cell relative to itself within its preferred behavior. So, of course, within a given session, a cell is going to have a perfect correlation with itself whenever the animal performs that behavior. But now, if you compare the tuning or the discharge rates, I should say, the deconvolved firing rates in session 1 versus session 2, you actually see a pretty similar diagonal going across here, indicating that the cells maintained the specificity of tuning, and they were even discharging to a fairly similar rate in session 2 versus session 1. If you compare the first section using the second session as the baseline, again, you see this nice kind of like red diagonal indicating that you have stability of tuning. Whereas if you make the same comparison, again, back to performance 1, if you make that same comparison for the observation conditions, the diagonal's not there, and there was no clear correlation structure that we could discern. And the same was true for M2. Now, this is the recordings from M2 that were all given basically the same treatment. So again, here's the cell that's tuned for grasping, that's not active during observation, that's tuned for grasping, but again, not during observation. And we made similar correlation matrices. It's the same type of comparison, where again, you see this the tendency for this red diagonal to persist across performance sessions, but again, it's absent during the observation session. So this is looking at single cells, and we didn't want to just limit ourselves. You know, one of the powers of using the mini scopes is we have lots of cells at a time, so we wanted to look to see if there was some sort of population structure that you just couldn't see at the level of single cells. So we did a dimensionality reduction using a uniform manifold approximation and projection, or UMAP, a pretty common dimensionality reduction algorithm. And we found that there were stable states in the recorded data for performed behavior. So I'll show you an example of that. So this is a performance session. Now, okay, so here's the animal. It's on a running wheel. In this case, it's running counterclockwise. In a second, it'll flip over and run clockwise. And here's the mini scope on the animal's head that's piping the data off. And all of those hundreds of cells we were recording were put through UMAP, and all of that activity was, the state space of the activity was projected down into a 2D manifold that's shown here as this gray circle. And so all of the possible state spaces during the recording are shown in gray. And the momentary state of the network is shown as the blue cursor. And what I want you to observe is that for as long as the animal stays in the behavior, the cursor tends to hover in the same position in the state space. So this is during counterclockwise running, it's in the lower left. Then the animal switches to clockwise running, and it just kind of hovers down here in the lower right quadrant. And it was kind of fun because you could even just look at where the cursor was on the gray map and just know if the animal was on the running wheel or not. So if you try the same thing during the observation session now, here is that same animal that was running on the running wheel. Now it's head fixed. We're recording the same cells, doing the same analysis, but again, it's during observation. And here's the demonstrator that's running on the wheel. And what you'll note maybe is that the cursor tends to kind of go all over the place, even though the animal's engaged in the wheel running in front of it. It's just not stable like it was during performance. Right, so we could quantify this and found it doesn't segregate perfectly, but there's a clear tendency for neural activity during certain behaviors to clump on the 2D manifold. Right, so this is running clockwise, shown here in beige. The green clump up here is turning counterclockwise. Here's rearing, shown in blue. Whereas, yeah, during the observation session, you look at the same cells, and it's like a Jackson Pollock painting or something that's just the dots are going all over the place. We made the same comparison. This is from another animal in N2. And again, here's running counterclockwise is fairly clumpy, turning counterclockwise. Here's eating, running clockwise. And again, during observation, the state space activity just didn't seem to be driven by the observed behaviors. And this is borne out by a pretty straightforward clustering index, the Dunn index, where you look at the inter versus intra cluster distance ratio. And we found that performance sessions were anywhere from 2.5 to 11 times more strongly clustered than during observation. So that was that. We next tried statistical modeling to see if we could model the cell's activity rate during a given behavior, if it was performed or observed. But we found, again, in every case, that the statistical model always performed better during performed than observed behaviors for nose poke, grasping, eating, grooming, turning, rearing. In all cases, the model performed better for performed than observed actions. And now the sort of counterpart is during the observation session, which is the stippled sort of bar graph. Down here, this is the model performance during each of the observed behaviors. Not so great, not really well able to predict the cell's firing rate based on the observed behavior. But whenever the animals would squirm, that was no problem. So we could predict squirming, as we called it, or fidgeting inside the tube. Whenever the animal moved, we could predict that just fine. And you can see very clearly in the neural activity too. So this kind of harks back to recent published papers from Anne Turkson's lab, as well as the paper with Carson Stringer and Marius Pachitariu, showing that if you have a head-fixed animal that moves, the brain really pays attention to that. And I think that's really telling us something. Right, so then if you look in M2, you see the same type of thing with M1, where performed behaviors really do a better job as predictors of neural activity than observed behaviors. The same is true for the observer's own movement. And finally, then there's the wheel running task, at least for PPC, as I'm showing here. Again, performance on the running wheel was a much better predictor, made for a much better ability to predict neural activity than observation of it. And again, there's the squirming effect. Right, so of course we wanted to know, since mice aren't little primates, they're not little monkeys, we wanted to assure ourselves that the animals were actually aware of what was going on around them. So to do this, we tried excluding periods where we thought the animals maybe might be paying less attention or were more out of it. And we used the pupil dilation as a proxy for this. So whenever the animal's pupil, whenever the observer's pupil was most dilated, we'd say, okay, this is when the animal's attentive. And based on our pixel intensity estimation in image J, that's what this trace here is showing. Whenever the pupil was dilated, it shows less negative numbers here for the black curve. Whenever the pupil was quite constricted, then we had more negative values here. So just arbitrarily drawing a threshold and cutting out the 10% of the session when the pupil was most constricted. We tried that. We tried to get rid of this low arousal period and see if that affected the data, but unfortunately it didn't. So if you look at data from the whole session, and this is data from 3 animals recording in M2, and the number of cells that had stable observation correlates was between 1% and 2%. If you include data from the whole session, whereas if you excluded the periods of lowest arousal, it was still between 1% and 2%, which is well below the false positive rate that we were calculating. All right, so arousal didn't solve it. And then we wanted to assure ourselves that the animals could actually see what was going on in front of them. So in this case, we had the animals in front of an empty box with no animal inside of it, but there were forceps placing food and removing it in front of them. And here's examples of 4 PPC neurons that were quite responsive. Whenever the forceps were appearing and placing and removing the food, the cells were active. And there was a subset of cells where we were recording that were strongly visually tuned to the presence of forceps, whereas other cells were not. But just based on the population vector activity, we could accurately decode just looking at the neural activity if the forceps were in front of the animal or not. So this is the actual decoder performance versus shuffle data shown in blue. So we were able to use the neural data to do quite a bit better than chance in this one simple visual test. And this was true also for another animal that we could use specifically for this condition. So then just to summarize, I'll summarize what I've shown and then just end with another task that we're using now. So what we found was that PPC and M2 were very strongly tuned to performed but not observed actions in the pellet reaching and wheel running tasks, which are just 2 tasks that we considered, right? Of course, there's other tasks that one could use. So it's possible that in mice, observed actions, are represented outside of the PPC M2 pathway, which kind of diverges away from what's known in other species, other mammalian species. But it's possible that actions, observed behavior is represented in higher visual areas like extrastriate areas AL or PM, or in cingulate or prefrontal areas that have also been studied in the context of social behavior. It could also be the case that detecting neural correlates for observed behavior in rodents might require that you do unrestrained social interactions, like what was found in Wei-Shi Hong's lab last year is one example of it, where mice were placed in a tube and they were doing a dominance test. Or using fear-based paradigms, like shocking the animals and doing social fear learning, because this is more basic to survival. And I know there's been lovely work from Kay Tai's group as well as Christian Kaiser's group more recently showing this type of tuning. So those might be more of the types of tasks to go after, perhaps, if you're looking for mirror cells. And we didn't give up on looking for neural networks supporting social learning in rodents. We really want to stick with sensory motor learning, just because I want to understand the visual motor transformation, how that happens in the brain, because to me that's still a complete and total mystery. So I want to stick with the sensory motor learning task here, which is what we're developing. And the key advantage here is that the behavioral learning, the perception of the action, is actually behaviorally demonstrable on the part of the observers, and both animals are behaving more freely and moving more naturally. And this is the PhD project of another student, Ida Rautio, who's been working very diligently on this for the last couple of years. So in the task, the animal taps ball number 1. This is the demonstrator animal on this side, and this is the observer on this side, right? So the demonstrator taps this ball, but nothing happens. It lights up. That's a cue. The animal taps it. It's just kind of a trigger. After the animal taps this ball, then this other ball lights up. The animal comes to it. When it taps this ball, though, it gets a very powerful rewarding stimulation by way of medial forebrain stimulation. And whenever the animal does this, the observer also gets the reward, right? So that incentivizes the animal to attend to what's going on here. So it's sort of like Pavlovian conditioning, where the CS, instead of being some neutral tone or a cue, is the behavior of this other animal. So you can do multiple sessions of exposure like this. And in this case, here's an animal that has had 5 observation sessions, and it's being tested for the first time. And it immediately started performing the task. So there's the trigger ball, and now it hits the reward ball, and it's getting the medial forebrain stimulation. And it's going to come back around, tap the ball, misses it, fixes it, and then gets this rewarding stimulation. So we are continuing to pursue this idea. And since the work is still ongoing, I don't want to show further behavioral data, but I look forward to being able to do that. So thank you very much for your attention. This is my lab, at least as of last year, and with Ida floating up on the top left. So again, I need to thank Tuğçe, Caroline, and Ben for their incredible efforts in making the mirror cell study possible. Bartul and Pranav did really nice work in helping put the paper together and performing analyses along the way. Ryan Cubero did the GLM and dimensionality reduction with our collaborator Yasser Rudi, also at the Kodley Institute, and Ida for her work with observational learning, and of course my funding sources. So thank you for your attention.
Thank you very much, Jonathan, for this great talk, and we will be looking forward to hearing more about your quest in finding those cells in the future. We have a number of questions coming your way. So the first one is, for the same animal, were the arousal levels comparable during performer and observer sessions?
Well, the arousal level during performance and observation. So I would think that during performance, the animals, it's engaged in doing the task. So if you're looking at pupil dilation, if that's what you're using as your proxy for attention, well, as animals move and are active, I mean, their pupils are just more dilated in general. So I think in that case, like necessarily, the performer, it depends on what you're using as your proxy for arousal. If you're using pupil dilation, it's going to be much more aroused, of course. And while we couldn't control for that perfectly in the observation condition, I mean, we tried, at least. So that's the best I can kind of answer to that.
Thank you, Jonathan. I have a second question for you. Does the internal state change the activity of PPC neurons? For example, hungry versus satiated mice in the context of eating or reaching for food?
Absolutely. So we wanted to keep. We didn't measure, or we don't seek to make claims about internal state is something that you can read out through activity in PPC, although it's an extremely interesting and extremely good question to ask. We wanted to keep the animals motivated, so both the observers and performers were on the same food deprivation schedule. and they were both trained, using the same types of pellets of food. And again, based on the tendency of the observer animals to react whenever the food was being eaten in front of them, we have to assume, at least to me, it's intuitively, I always thought it was pretty clear that the observer animals were also hungry, and that when they couldn't get the food, it drove them a little bit crazy. But the idea of looking at internal states and how it affects population tuning to actions, I think, is a fascinating question. We did not get at that here, though.
Okay. Another question. Have you thought about looking at different brain areas for observation? Is it possible that the neural basis for imitative behavior is the result of a network activity rather than a very well-defined pop of multisensory neurons? Have you tried to analyze the neuronal activity looking for clusters of cells active in a given time window rather than individual cells?
Yes. So number one is PPC and M2 were areas that we picked it was motivated by the primate literature. And again, it's entirely possible that the types of things that we were wanting to see or hoping to see could be just ventral, down in cingulate cortex, or in prefrontal areas. That's entirely possible. And so I wanted to be clear in my talk here, as we were in the paper that was published a couple of months ago, that absence of evidence is not evidence of absence. So we can't rule out or claim to disprove that mice have neural cells. They can certainly be happening in somewhere else, and they could be happening in the context of a different type of behavior. And in terms of looking for activity of multiple cells instead of single cells, we tried that. So we tried just looking. We took behavior, population activity during the performance session, and performed affinity propagation, which is just a clustering algorithm that actually succeeded in pulling out patches of neural activity, kind of like what you saw in the UMAP results. It tended to pull out patches of coactive cells during performed behaviors, but again, we just weren't seeing it. It was just absent there during observation. But that certainly was the dream at the beginning, was to not even have to do behavior labeling and just let the neural activity tell us when something interesting was going on. And that tended to happen when the animals did performed behaviors, but we just didn't see it during observed behaviors. So.
Thanks, Jonathan. So before we pass to the next section, I just wanted to announce that we also have Kunal Ghosh today, the CEO of Inscopix, and he would like to give a short acknowledgement speech. So I'm going to mute myself and ask Kunal to unmute himself.
Thank you, Fabrizio. Good evening, I guess, to most of you. I believe most of our attendees today are based in Europe. Thank you very much, Jonathan and Yaniv. It's really nice to see you both, virtually, albeit, but really grateful for both of your participation today in making this webinar possible. Appreciate the talks. I had the good fortune of listening in to the last part of Yaniv's talk and to the last part of Jonathan's talk, and really enjoyed hearing the summaries of the work that you've both been doing. So thank you again. Our sincere gratitude to both of you. Thank you for helping keep the neuroscience community connected, especially the European neuroscience community during this time of crisis. Hopefully, with summer arriving, we are all quite optimistic that the worst of the crisis is behind us, and we'll all be able to visit each other and start, again, connecting with each other physically and in the traditional means that we have been used to networking in science and beyond. That's all I wanted to say. It really was my intention today to just thank the speakers, to thank the attendees, and we certainly look forward to getting together soon in Norway, and in Israel, and in other parts of Europe. We hope to continue having such seminars, not only virtually, but also physically as the world starts to open up. So thank you again, gentlemen, and thank you to all of you who made time today to attend this virtual webinar seminar. Thank you, guys.
Thanks, Kunal. Thank you. Now we pass to the next section of the webinar. We got Jonathan Zapata, our field scientific consultant, that would like to share some slides with us. Thank you, Jonathan.
Can you see the presenter mode now? Just
yeah, we can see your presenter mode.
It's okay.
Yes, perfect.
So hi everyone, my name is Jonathan, and I am a field scientific consultant, a senior field scientific consultant at Inscopix, covering South Europe and Scandinavia. So first of all, I would like to thank Yaniv and Jonathan for the great talks and discussion, and thank you all for attending our first European webinar. And I would like to take a few minutes to give you some information about the Inscopix support. So the field scientific support and the field scientific consultant team at Inscopix is a group of experienced neuroscientists that provide expert level training and consultation on the application of miniature microscope technology to promote the rapid advancement of neuroscience research and understanding of the brain. So we will use this background and additional extensive and Vista-invoked in-house training to provide support that will help your lab succeed in getting data from your system all the way to the publication process. So this support includes, but is not limiting to, inputs on experimental design, behavioral setup synchronization with external devices such as video tracking or EEG, advice on viruses, transgenic lines, and lens selection. We will also provide on-site visits and guide you through the surgical procedure to optimize the optical access in the targeted brain area and assist you during data acquisition and primary analysis using our Inscopix data processing software, IDBS. So this support is not limited to imaging success and will help you with manuscript, figure preparation, and grant proposal if needed. Our European FSC team is composed of Diane Dames-Verneau, Giorgio Lizzi, and myself. So please feel free to contact us if you have any questions. You can even ask questions in the chat or contact us directly using the following email address: support@inscopix.com. I hope you had a great time with us, and I will leave our EMEA sales manager, Fabrizio, say a few words and close the webinar. Thank you so much.
Thank you, Jonathan. And just, can you unshare the screen for me?
Yes.
Thank you. So, well, first of all, I would like to thank Yaniv and Jonathan Whitlock for giving us a great talk about their most recent data. And I would like to thank all of the people that registered and attended today's webinar. I hope you enjoyed it, and I'm looking forward to having you again with us on the next Inscopix webinar. Thank you, everyone. Bye. Goodbye.